Fact of the day.
Hello, hi, and welcome to Fact Today with me, Mark Lotsu. So today's Fact Today, not about penises, so don't worry about that. It is about lots of response for the old penis one, but that's good. This is about a serious medical topic. It's talking about cancer today and the evolution of cancer. So normally, what happens is you've got two main groups of genes which control whether or not cells can then become cancerous. You've got two tumour suppressor genes which stop the cells from dividing lots and lots and lots to form tumours. If they mutate, then the cells divide lots and lots and you form a tumour. And you've also got genes called proto-oncogenes. And proto-oncogenes promote cell division for growth and repair. If they mutate, then they can divide even more, and then you could develop a tumour. So how do we diagnose a tumour? So normal way, you can use X-rays, you can use CT scans, you can use fMRI, you can do all of these to kind of see if there's something abnormal there. The only way to actually truly say yes, this is exactly a tumour, this is cancerous, is by taking a biopsy. A biopsy is a sample of the tissue that you remove and you look at under a microscope and say, oh yeah, these cells are cancerous. Biopsies are highly invasive. If you've got leukemia, if you've got a lymphoma, you actually need to go into the bone marrow, syringe into your bone marrow to take a sample out. That's hardcore. If you've got a potential cancer in your brain or spine, then you need to get into the cerebral spinal fluid. Again, this stuff is not nice at all. Then in terms of the treatment, you've got, you can cut out the cancer, you've got chemotherapy, you've got radiotherapy. What the issue is that you've got what's called a trunk tumour and then you've got branch tumours. So the trunk tumour is obviously the main bit, how it starts, and then you've got branch tumours. Now cancer cells can evolve, they can mutate, just like bacteria do with antibiotics. You know how we've got bacteria resistant to antibiotics. Cancer cells can do exactly the same thing. And when cancer cells mutate, they change, the marks on them change, they're no longer affected by certain chemicals, they may be affected more by other chemicals. So what you have to do is you have to take a biopsy each time you want to see if there are new cells there. That's massively invasive and really, really difficult to do, and a lot of people don't want to go through the pain of that operation. So I've now got a method called circulating tumour DNA, ctDNA. So when a cancer cells die, the DNA is released into your bloodstream, travels around your blood, you take a sample of the blood, you can look at the DNA and say, right, this cancer is now mutated to this form, we now need to use these chemicals, these drugs. Perfect, that's how it works. It's amazing, it's so good that obviously biopsies are horrendous and this is a much better way of doing it. That's Fact of the Day. See you later. Love you. Bye. Love you. Bye.